It's a real pleasure to have Janine Mendola here. Thanks for coming. So Janine actually did her PhD together with David in Boston. We heard already a few stories about that over lunch. And that was, she did it in with patients, so really like sort of neuropsychology. Right during their PhD, fMRI was invented, and that was a technique that she got very excited about and jumped onto for a postdoc and never left it since. Hello. And now she's a professor at McGill University already since 15 years, still working on visual system, visual perception, illusions, and well, I'm very excited to hear what you're going to tell us.
Thanks, Mary. Hello. Thanks for coming today. The sound is good for you? Okay. So today I'm going to talk about my favorite visual illusion for sure, binocular rivalry. I suspect most of you are familiar when you show different images to the two eyes. This results in perception alternating between the two possible images. I like to say that rivalry is just one of many regimes, computational regimes that the brain can use to integrate the images shown to two eyes. But in rivalry, we believe that both interocular inhibition, i.e. competition between monocular input, as well as competition between higher level binocular percepts, likely is occurring in most cases. So binocular rivalry is a prime example of bistability, where conscious visual perception changes without any change in the retinal image. And so this is quite attractive for studies of physiology of the brain, so that we can monitor sort of the intrinsic functions of cerebral cortex while the early stimulus never changes. So my plan for the talk is to just introduce binocular rivalry a bit further and to establish the fact that there are individual differences in the speed of binocular rivalry. And then I'm going to focus today on my MEG experiments. I certainly have done fMRI in the past with binocular rivalry, but I have so much fresh new MEG data collected from a relatively new center that we have at McGill. So that's what I'd like to share today. So I'm going to talk first about studies in which we tag the visual images, and I'll explain what I mean by that. And then talk about some untagged experiments where we allow rivalry to be occurring more naturally. So I'll be talking about MEG measures of power, coherence, functional connectivity. So I thought I would just acknowledge that probably behaviorally in a psychophysical paradigm, the most dominant paradigm is to study stimulus-dependent effects within an observer. But it's equally possible to study observer-dependent effects for the same stimuli. And of course, that's what we are talking about when we're talking about individual differences. And it turns out that the rate of alternation between those alternate dominant percepts does vary in a reasonably stable way across people. So slow, this typically ranges from every second or so for a fast switcher to every few seconds for a slow switcher. And it's nice that twin studies have even established that this is a partially heritable trait. And just a little bit more about this trait-like aspect. It's consistent that it's been shown that rivalry rate can vary across groups, in terms of conditions such as autism or aging populations, or even medications and other changes like that. I also want to point out that models of binocular rivalry generally emphasize reciprocal inhibition between two separate pools of neurons that are competing for access to awareness and represent the alternate perceptions, or percepts, I should say. And in general, the balance between excitation and inhibition is thought to be important for this to manifest. So I simply want to acknowledge in a basic way, like the most sort of basic kind of model that most people have of rivalry in their heads in an area like V1 would start with monocular input coming from each eye from the LGN, and orientation selectivity would allow neurons with opposite orientation preferences to be reciprocally inhibiting each other in a cyclical way that will given neuronal adaptation will change over time. There's likely to be reciprocal inhibition happening between binocular neurons as well. Now, these models can get quite complicated. There could be multiple sort of recurrent loops of inhibition happening here. There could be feedback effects that are relevant. So it's a complicated system for sure. But this is kind of a basic model that we have in our head. And it also allows me to sort of introduce the idea that when we're doing MEG or EEG experiments, there's a technique that's been around for quite a while. It even has some sort of theoretical underpinnings, kind of from systems-based engineering kind of principles, where we can take the two images in each eye and we can flicker them at a distinct rate. So there is a different flicker in each eye. And by the way, that alone does not cause binocular rivalry. But the nice thing about having these different flicker rates is that it allows us to follow the presence of the input frequencies deeper into the visual system in terms of the evoked response that we measure over cortex. So these frequencies are referred to as the tags. So, and in fact, if you're looking at a sort of basic representation of MEG data that plots the log power against the frequency of the signals, you get sort of a 1/f fall off in general of the signal magnitude. And if you're tagging, it'll be evident to see peaks in those tags frequencies right there. And we also plotted a separate data set where we have rivalry that is untagged without the flicker. So I'm going to talk for a minute or two about an older study that has been published where we contrasted several different stimulus conditions using this tagging approach. So we had three conditions. We had the binocular rivalry. We had another condition called pattern rivalry, which I'll show you in a second because it kind of makes for a nice demonstration. And then we had a non-rival risk condition just as a control. And if you've ever seen pattern rivalry, you may be familiar, but if you stare, if you fixate reasonably well in the center of the image, it's pretty easy for most people to see that the red or the green gratings or bars will sort of shift in their sort of salience, in their perceived depth order. And so that works pretty well. So this was something we, in this particular experiment, we used this as a control of sorts because it had all that rivalry mechanisms were still at play, but without the interocular competition. We have really new data where we have actually tagged the red and the green components separately, but that's for another day. So in this experiment, we had 10 subjects. This is an older data set. We only had 10 subjects, but we were doing dicoptic flicker, and we had a photodiode to record the stimulus flicker and its phase so that we could filter the evoked responses. And we used prisms to provide the visual input, and subjects are responding with a button press to let us know which stimulus they see. And in this particular experiment, we also tried to take some account of something called mixed percepts, which are pretty interesting. So I'm going to take a moment just to acknowledge this concept of a intermediate state that subjects often traverse when they're going from, say, red stimulus dominant to green stimulus dominant. There is often a short period of time of intermediate perception. And if in the literature on binocular rivalry, if you go back more than, say, 10 or 15 years, people hardly talked about this mixed percept as a concept. People were just given two buttons and they were told, just make it happen. It's either mostly one or it's mostly the other. But in recent years, people have started to take this transition more seriously as an interesting state. And I've also found evidence to support the interest of this idea. So the MEG methods here are pretty standard. I don't really think I need to dwell on them. We had a 275 channel system. We're deeply indebted to the wonderful software program called Brainstorm that has been sort of written and promoted in part through the Sylvain Ballet, who runs our MEG Center. We're taking advantage of having MRI images of our subjects to have more accurate source localization. And we already talked about the tagging at the fundamental frequencies. And I forgot to mention that there are also harmonics that are often detectable when you're doing this tagging. And so sometimes these intermodulation frequencies are referred to, which could represent, say, the sum of the two fundamentals or the difference. And so we ended up finding some interesting data that related to the difference of our two fundamental frequencies. So as I said, we have slow or fast switchers. And for the sake of a couple of the slides that are coming up, we ended up separating our subjects into two groups according to whether they were the more slow or fast switchers. And so here's, we finally get to look at some data. And this is certainly standard, and I'm definitely not the first person to show this, but it's nice to be able to see time zero here. This is an event-related plot. Time zero is when the subject pressed the button to say that they switched percepts. And so it's quite a robust finding to find that the power of the stimulus that is now become dominant will peak, and the tag associated with the now suppressed percept is diminished. And so you see this waxing and waning of the power at the fundamental monocular frequencies, although these signals are likely being driven by both completely monocular neurons as well as neurons that have some binocular integration as well. We have the PR condition is just serving as a control here. There's no interocular inhibition at all. So we have a pretty nice source localization that we can get with these modern MEG methods. And we find that there's a pretty wide expanse of visual areas that increase the power for the dominant tag and decrease in power for the suppressed tag. We have here one of the atlases for retinotopic areas here. The Wang et al. Atlas is here. V1 is shown in a bright green here. And there's a really quite robust difference in many areas show a robust difference between the dominant and the suppressed power. And there's much, much less systematic variation for the control. I should have said that this data is being plotted at minus 300 seconds here. So now I'm showing you exactly the same data that we just saw, but I'm just going to sort of chop up the pie a little bit differently and show the dominant and the suppressed traces again, but now for the 5 faster switchers and for the 5 slower switchers. And I just basically wanted to establish this impression of the slow switchers in dashed lines and the fast switchers in the solid lines. It's pretty similar. It's actually a little bit intriguing to see more of a dominant signal for the fast switchers, but that particular effect was not significant. But nonetheless, here's the brain map showing a little more impressive dominant signal for the fast switchers than the slow, but otherwise pretty similar, actually. And the next slide was the effect that we found that was a more impressive difference between the fast and the slow switchers. Was we actually found when we looked at that intermodulation frequency. So we saw a clear peak in the power at that difference frequency, really just for the slow switchers. And now we're coming back to that idea about the mixed percepts in between transitions. So our design had some limitations here, but we were able to sort trials in which there were mixed percepts separately from trials in which there were not mixed percepts included. So what we found was that only for trials that included mixed percepts and only for the slow switchers did we find this particularly robust power at the intermodulation frequency. Yes, and so I'll come back to that at the very end. This difference frequency is a little bit interesting, in particular, because there are some theoretical models of binocular integration that posit that there might be neurons that actually do code the difference between the left eye image and the right eye image, which is actually kind of I think it's pretty intuitive to imagine that that difference between the two images would be a pretty efficient coding of the left and the right eye image. But actually, historically, in visual research, there hasn't been a lot of evidence for neurons that actually code the difference between the two images. There have been other approaches like correlation with Bello-Ulesh and things like that. There have been the field has gone in a different direction, but there are some researchers who are still quite devoted to the idea that there might be neurons that code the difference between the two eyes, that like they would be opponency neurons for the two eyes. And so this could be consistent with that. But finally, following up on this idea of binocular rivalry rate as a trait like feature, we wanted to continue to look at the relationship between this intermodulation frequency and rivalry rate. And so we did a whole brain linear correlate regression between the durations of the dominant percepts against the power at the intermodulation frequency. And there was actually sort of a network of areas beyond V1 in which we could predict rate based on the power of that frequency. I think I'll leave it at that. There are interesting members of this network, I guess you could say, like the retrosplenial cortex, lateral occipital region, which is quite interesting for sort of mid-levels of vision. Superior parietal cortex having certainly probably a strong role in modulation of rivalry and maybe in terms of initiation of switches. So my interim summary here would be that we observed the eye-specific tags to wax and wane in tandem with rivalry, and we extended the finding far beyond V1, which has dominated the older literature. And we linked that intermodulation frequency with slower switchers and mixed percepts. So this could at least be consistent with models of ocular opponency neurons, efficient coding. And it sort of might be suggestive of the idea that for the slow switchers and mixed percepts could be a time of sort of quite high inhibition, or quite high competition between the two alternatives. Okay, so I think I already talked about different areas in that network in which we could predict rivalry rate based on that intermodulation frequency, and particularly with areas like the retrosplenial cortex showing up. It was kind of fun to speculate that this network might deal with conflict in an increasingly abstract way. And in general, this network also overlaps with the default mode network, which is associated broadly with both normal and abnormal interoceptive integration and monitoring. Okay, so now I'm going to show you yeah, of course. Absolutely. Yes. It's a dynamic percept. It would definitely be changing over time. And for all I know, it might differ slightly between observers. I can't speak firsthand, but I just want to establish that it's very rare for subjects to see plaids. So that's not really what we're talking about, which is why rivalry is eminent here, right? Only for very, very low contrast would subjects typically ever see a plaid. Instead, they're seeing essentially patches, which may sometimes be evident to have a little bit of a wave-like property, was sort of start in one side and sort of grow to the other. But patchy, yes.
So from time to time, it looks as if like a patch, like let's say if you have two orthogonal orientations, like you will have some patches of different orientations.
Yes.
And then you will dynamically change.
Yes, or
while until it's settled with 1%.
Correct. I think what you say is all correct. I might just also add that in my experience, it would also be quite common to just sort of to have the suppressed percept sort of pop up, say, at one side of the stimulus. So in that sense, there's just one patch, and then sort of like grow to cover the remainder of the stimulus.
And another question is, you showed that inflated brain, the MEG result, like suppression. Can we go back to that?
Of course.
Yeah.
Way
earlier.
Like way in the beginning?
Yes, that one. So it looked as if in Binocular Rivalry, B, the second one, or the first one, there's quite some suppression, and it's dark blue. Is that region, that's something like LO right there?
Yes. I think LO is
somewhere
probably right there. Yes.
I see. But the most saturated one is right in between the visual areas. What is that area?
That's
right.
Yeah,
that
that's a fair point. This is sort of, I think, like upper or lower STS here. This is fusiform, I think. Yeah, I haven't thought about that in a long time. In a long time. I mean, I must say, we have, obviously I could make plots like this. I generally show V1, but of course we can make plots like this for any one of these regions. I would have to say that for the most part, they look fairly similar. There might be subtle, interesting differences, but I don't really have any basis to imagine that this area here is going to look radically different from the neighborhood. But yeah, I mean, I hadn't noticed that in a long time. Yeah.
And one last question.
Yes.
I'm sorry, but I didn't quite understood the tagging part, like 5 hertz, 7.5 hertz
Yes.
tagging. So it means like you have one stimulus and then you turn it off, or what is that frequency? Like it's kind of flickering?
So yes. So imagine that I have a stimulus on the left and the right side of the monitor, and one of the stimuli is flickering quickly at 5 times a second, and the other one is flickering at 7 times a second, let's say.
What is the on duration and off duration for, let's say, 5 hertz flicker?
In this case, I think it was, well, actually, okay. I think for this older data, I think we're not counterphasing or anything like that. It's just stimulus absent or present.
Okay.
Okay.
So it's like box call, like on and off. It's
Yes.
not sloping on.
Nothing fancy.
Okay,
No.
I see. Got it. Thank you.
Yes. Okay, so this is the same data now, but I'm just going to analyze it with a slightly different metric now. And so coherence is kind of a cool metric, because now we can be sensitive not only to the power of those fundamental frequencies, but also the relationship of the phase and how consistent the phase of those the phase of those signals are compared to the source of the flicker, which is the stimulus itself. So we can look for evoked responses that are in a precise phase relationship to the stimulus. And in general, we're going to be more sensitive here with a coherence analysis to synchrony, essentially. Okay, so we'll be giving a boost to signals that are in strong phase alignment with each other. So this is the same data. I think in this case we actually used a different control condition here. Now we're just using the non-rivalrous control, but here we are now plotting coherence. Yes, this should have said coherence amplitude. Sorry, it's been z-scored. But once again, we see the coherence is waxing and waning in concert with the suppression. So it's very similar to power. But when we looked at it a little more carefully, we found it quite interesting to notice that there seems to be more of a delay, or a time delay, between the peak of the dominant coherence and the trough of the suppressed coherence. And actually, this is pretty interesting for people who really think about how rivalry would be modeled, because there are 2 possible regimes that have been discussed. And the one that the data supports here is called the escape theory. And so basically, this is just saying that rivalry, or let's say a switch from one percept to another, is going to occur when the dominant signal increases to a certain trigger level, okay, rather than the alternate situation of say, a switch occurring when the suppressed, reaches a low enough value. So it was quite interesting to see this, because it's consistent with this escape theory. And there's actually some aspects of psychophysics, which I won't drag you through, but there's some interesting aspects of psychophysics that are also consistent with the escape theory. Like, for example, you give the stimuli more contrast and you generally increase the alternation rate, and things like that are actually sort of consistent. Yeah. Right. Do I have that here? I don't actually think I have that slide, which is funny because it's a figure in the paper that we published. Maybe slightly. I'm going to address your question in kind of a couple different ways, but it's a good question. But before I do that, let me just say that we wanted, after seeing that observation, we just wanted to address that a little bit more systematically and to see if we could find any individual differences. And so, and we ended up deciding that measuring the slope of that dominant percept, like the slope of that red trace, it's a little bit more of a stable thing to measure than the time to peak. And we did find that the slope of that dominant percept, has a modest correlation with rivalry rate, or how long people typically have their dominant percepts. And it's nice that we don't see that relationship for the suppressed trace. Okay, so just interim summary here. We just talked about how the dominant coherence peaks earlier. It's consistent with the escape theory. And actually, there's other examples in vision science where we see that delayed monocular signals may be adaptively suppressed, or maybe maybe delayed signals in general might be adaptively suppressed. It's kind of an interesting concept. And actually, if you think about it, the theory is also kind of makes sense in a natural setting where it could accommodate the need to attend to a new event, of sufficient urgency by responding to a potential, with a potential shift in awareness. Okay, so far so good? Okay. So I'm going to talk now about, briefly, I'll talk about an analysis of connectivity, which would kind of address your question a little bit more. So there are many different ways that you can try to calculate connectivity, a lot of different metrics out there. And we used one called phase transfer entropy, which is well integrated in Brainstorm software. It's essentially it's in general working on the same principles as, say, Granger causality or any of the time delay based measurements of connectivity. We picked a few regions of interest that were independently defined from another subtraction. And so this is the data. So these matrices are a super efficient way to show you the data, but they're very hard to read. So, we're plotting, areas where the connectivity between two areas is stronger in one, between a pair of connections is stronger in one direction versus the other. And the asterisks show which is significant. But if you trust me, I can tell you that all of this just basically, is a diagrammed here. Now, one of the things we saw right away was that all of the connections that are significant here are feed forward. And we think upon, a moment's thought that is almost certainly due to the tagging that we're doing. And we have some other untagged data where we are also able to see some feedback connections now. So this is not a functional connectivity that I think would represent rivalry in a more natural setting. But it was still very interesting to see that we could have a different signature of connectivity for the dominant percepts compared to the suppressed percepts. That's a pretty new topic in the literature. For a long times, I think you could say that dominant and suppression were just sort of considered symmetric, kind of like by default. But I'm increasingly interested in, looking at rivalry and asking, or considering the possibility that a switch to dominant may be fundamentally different from a switch to suppressed. So I'm interested in any way that I can dissociate the two. And so we see a slightly different pattern here. Interesting to see a kind of a strong connection from V1, V3, maybe I would imagine ultimately involving the parietal cortex insofar as the connection I made between dominant percepts and alternation rate. So we found some distinct connectivity patterns for dominant compared to suppressed. And in this case, it was all feed forward type. And I could say that the broad association that I made in this paper between parietal regions and dominance on the one hand, and ventral regions with suppression on the other hand, is kind of consistent. There is this broadly consistent in the literature. But this is one of the first attempts to actually dissociate dominance from suppression, and not just to consider them as sort of symmetric or anti-symmetric in a way. Okay. Well, I could complement those results by just talking a little bit about untagged rivalry. So now subjects are just doing the same rivalry task with the MEG recording, and we're simply not flickering the stimuli. Okay. We made a few small changes because this was a new data set. So we were able to get a larger N, which is nice. So now we have 25 subjects. We used a slightly different regime for the key presses. We had a key for one stimulus, another key for the other stimulus, but subjects were told to press the button for the duration of the dominance and then to release to indicate mixed percepts. Yeah, and we have typical behavior here with a mean dominance duration around 2 seconds. And now we're actually explicitly measuring the mixed percept duration because we were really getting more interested in that, and sort of a typical range of rates. And for this afternoon, I'm just going to talk about alpha as one of the more obvious things that we could look at in our untagged data. But we're going to look at event-related alpha. This is just the paradigm I just explained in terms of pressing down one of two keys for dominant and releasing for mixed. Now, for some of you, this might be old hat, but alpha oscillations have been of interest for quite a while. There's a review paper, which I think is quite nice, that does a pretty good job of just trying to organize all the different conceptual roles that alpha might play based on, the rather large literature out there. And so I'm going to focus mostly on the idea of alpha as an inhibitor or alpha as a stabilizer. And we're asking if alpha can support the inhibition of the suppressed percept and or the stabilization of the dominant percept during rivalry. In the literature, there was one relevant paper already published using EEG. They don't use binocular rivalry. They use a Necker cube to induce the bistability. But this is an interesting paper because just like we might expect, if alpha is a stabilizer, they found a decrease in alpha immediately preceding an alternation in a parietal occipital area. And indeed, the power in POS after the emergence of a new percept predicted its duration. So we're left with this idea that high alpha promotes stability and suppresses switching. And so our MEG results are consistent with that. We're able to do this event-related So time 0 is once again the button press. What we did here, kind of for fun, because we could, we have done tagging in the same 25 subjects as well. Okay. And so we thought it was quite interesting, even though these are different. This is a different data set collected on 2 different days, but the subjects are identical. So we're sort of taking data from 2 different days and overlapping the tagged in red and green along with the untagged data. And everything has been event related to the button presses. So just to kind of give you some context for the alpha power here, sort of decreasing right before a switch in percept. Of course, we know we can plot that for the whole brain, and you can see it's actually quite widespread reduction in alpha right before perception switches. Now, I can't resist showing you in passing that we actually find the opposite pattern for these mixed percepts. So we actually find alpha is higher right before a mixed percept, which could really support the idea that these mixed percepts are a time of a very strong competition. In which a sort of suppressing a switch. Just for fun, I just have a little movie here to show you the alpha unfolding. And I thought this had value because I think it's quite evident that you can, at least at the level of our source localization here, it's pretty evident that the alpha power seems to be sort of a feedback. You can see it starting early in the parietal regions, and also a little bit in sort of ventral temporal, and sort of feeding back to V1. It's pretty easy to see that here. And this is all right before the switch. And then it starts to go back from where it came. So there are many ways to analyze this data. I mean, just as a visual image, I'm just pointing out that, of course, you know, I show you these plots in V1, but as you pointed out in the audience, I could plot these, make alpha power plots for lots and lots of regions of interest. So this is a very rich data set. We're in the process of still analyzing this. You know, it might lend itself to multivariate methods. So we're looking maybe at some multivariate analyses of that type of data. But just a preliminary analysis that we've done on connectivity here, I thought would be relevant to the story that I'm telling. This data here is actually just a preliminary analysis that we did using Granger connectivity. And we intend to follow up with the same PTE connectivity that I already talked about. But just, as I said, this is just preliminary, plotting the top 5% of the Granger connections. So it's consistent with our speculation that when we tag the stimulus, we seem to have a stronger bottom-up influence from V1, say, to areas like V3, and all sorts of other interesting things are obviously going on here as well. This is, as I said, it's just preliminary, but in the untagged rivalry, now it's of somewhat encouraging, I think, to see more top-down influences are now measurable by us as well. That's a great question. I've been interested in LO for a long time in my career, even going back to doing fMRI studies of illusory contours, actually. So LO is one of my favorite areas. LO certainly shows up in this preliminary analysis, just not in the top 5%, that I'm just kind of showing here for fun. I'm doing another experiment. I don't want to get too far off topic here, but I'm doing another experiment, which sounds like you might be interested in it because you were quite interested in the piecemeal aspect of mixed percepts. But there's a type of rivalry where people purposefully take an image like these gratings and just imagine chopping up the image into a few different pieces, like a quilt or something like that, and then distributing the pieces of, say, the red and the green image in different eyes, right? So or to keep it simple, we just chop the stimulus down the middle, and one eye sees green and red, and the other eye sees red and green. Okay, so neither eye sees the entire image, but together they can integrate the image. And it's fascinating that rivalry is very robust to that type of manipulation, which is a really good sort of like prima facie kind of evidence that this cannot be accomplished just with monocular neurons, because they would never be able to integrate that. And I'm not talking about that data today, but in that data, we have seen like LO sort of popping up as maybe being particularly active when that integration is occurring for the interocular grouping, as we call it. So, okay. So far, the data definitely supports the hypothesis that high alpha promotes stability and suppresses switching. And in V1, we see that alpha dips prior to a switch to the dominant percept, and that alpha peaks prior to a switch to mix. And so far, the data is suggestive of a modulatory feedback mechanism, which we would have expected, say, from the literature, due to, say, TMS can disrupt rivalry when placed over the parietal cortex, for example. Okay, so I have overall conclusions here, and I'll probably stop here. So overall, binocular rivalry is a powerful platform for the study of visual awareness. Our data suggests that a wide expanse of posterior visual cortex participates in stimulus selection, that coherent parietal occipital connections help to dictate the current perceptual interpretation in concert with conflict management. The interplay of excitation and inhibition between complementary neural networks may even serve as a model of brain processing in general. I mean, I love binocular rivalry. Of course, I'm kind of biased, but I think it's totally rational to imagine that this kind of competitive interaction between neural networks, is happening all the time between a wide variety of networks. So, and it's also quite interesting to me that some of the relevant variants here can certainly be attributed to some stable trait-like features. We can predict rivalry rate based on various metrics during tasks. And we can also predict rivalry rate based on some features of resting state MEG data as well. I have a few more slides where I could talk about that if anybody was deeply interested in that. We've been looking mostly at power in the alpha band again, but we can predict who's a fast switcher or a slow switcher based on aspects of resting state data that could be easily interpreted according to, say, balance of excitation, at least local balance of excitation and inhibition, like, say, in an area like V1. Those of you who are really into MEG and are familiar with the 1/F power drop and the way that those sorts of data are sometimes interpreted in terms of neural noise. So some of the data that I'm not going to show you, I don't think directly, where we can predict rivalry rate based on the exponent of that drop in resting state data. So you could even potentially interpret that along with the idea of noise, because people do like to include noise when they model binocular rivalry as well, because it's the adaptation over time along with kind of the inherent noise of the neurons that kind of keeps them from ever settling kind of like, stably for just one percept forever. So you sort of need adaptation and you need noise. And so in theory, some of these resting state features might be related either to the EI balance or in theory, maybe to some meta measurement of noise. So we can also predict rate based on some resting state features as well. So thank you so much for your attention. And yeah. Let me just acknowledge my wonderful collaborators, my own student, Eric Mokri, and some people who've been working either in the past or the present with Sylvain Ballet, who leads the MEG effort at McGill. He's really a great colleague. So thanks.